Hello, we're glad you've joined us for this live webinar, Generation of Purified Human iPSC-Derived Cardiomyocytes Using Clinically Relevant Workflow. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Miltenyi Biotech. Miltenyi Biotech is a global provider of products and services that advance biomedical research and cellular therapy. The company's innovative tools support research at every level, from basic research to translational research to clinical application. This integrated portfolio enables scientists and clinicians to obtain, analyze, and utilize the cell. Miltenyi Biotech's technologies cover techniques of sample preparation, cell isolation, cell sorting, flow cytometry, cell culture, molecular analysis, and preclinical imaging. Their more than 25 years of expertise spans research areas including immunology, stem cell biology, neuroscience, and cancer, and clinical research areas like hematology, graft engineering, and apheresis. The company also offers comprehensive scientific support, consultation, and expert training. To learn more, visit www.miltenyibiotech.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Todd J Herron, BS, PhD, Director of the Frankel Cardiovascular Center's Cardiovascular Regeneration Core Laboratory. Dr. Herron is on the faculty in the University of Michigan Medical School and has appointments in the Department of Internal Medicine and Molecular and Integrative Physiology as an associate research scientist. His research is focused on the complex interplay between cardiac electrical excitation and contractile force generation, a process known classically as excitation-contraction coupling. Excitation-contraction coupling is highly regulated in the mammalian heart, and slight modifications of this process occur on a beat-to-beat basis. Any dysfunction can lead to fatal cardiac arrhythmias and heart failure. In this research, Dr. Herron has utilized transgenic animals, surgical models of heart failure, and more recently, human-induced pluripotent stem cells, hPSCs. Dr. Herron's lab was among the first to utilize hPSC-CM monolayers to study human cardiac arrhythmia mechanisms in vitro. Current research is focused on developing patient-specific cardiomyopathy disease models in vitro using hPSC-CM technology, as well as development of high-throughput screening platforms to test for compound effects on human cardiac function. A primary goal of the core laboratory directed by Dr. Herron is to work toward generation of clinically relevant hPSCs and hPSC-CMs by using xeno-free media and non-genetic modification-based purification of derived cells. In this endeavor, his lab relies on clinically relevant reagents from Miltenyi Biotech. Dr. Herron's complete bio is found on the LabRoots website. Dr. Herron will now begin his presentation.
Thank you, Judy, very much for that introduction. I'm happy to present work that I'm doing now in my laboratory to create unlimited supplies of human cardiac muscle for research purposes. So in the Cardiovascular Regeneration Core Laboratory, we obtain skin biopsies from patients, and through the process of cellular reprogramming, we create unlimited supplies of patient-specific stem cells, which then enables us to create unlimited amounts of patient-specific cardiac myocytes and tissue. And we use these cells to work on drug discovery projects, as well as to determine novel mechanisms of disease. We use platforms such as two-dimensional cardiac monolayer optical mapping and arrhythmia study to determine cardiotoxicity. We also utilize three-dimensional organoids to study structure and function, as well as contractile function of human heart muscle. And our overall goal is to utilize this technology to create new and safe therapies for patients. In our endeavor to create clinically relevant cardiovascular cells for research purposes, we utilize an array of products from Miltenyi Biotech. One product that we use extensively is their pluripotent stem cell medium, which is called StemMax iPS Brew, and the XF stands for xeno-free. This enables the generation of induced pluripotent stem cells in media that is free of animal products. We also utilize their monolayer dissociation kit for human tissue, as well as their newly developed PSC-derived cardiac myocyte isolation kit, which is optimized for human cardiac myocytes. Now I'm going to show data which supports the successful reprogramming of human dermal fibroblasts using sendivirus in media that is the iPS Brew xeno-free media. And in this first slide, I show results using a reporter virus that carries green fluorescent protein. And here you see successful transduction of dermal fibroblasts with sendivirus in the iPS Brew xeno-free media. This plot shows time-lapse imaging studying the appearance of GFP expression in dermal fibroblasts in the iPS Brew media. In the next slide, we show successful reprogramming of human dermal fibroblasts to hPSCs in iPS Brew. This shows time-lapse imaging starting from day 0, where there are dermal fibroblasts in culture, and by day 15, we observe very robust and very efficient generation of hPSC colonies in iPS Brew. And the way that we monitor the time-lapse imaging is by imaging these vessels at 2-hour intervals in the incubator using an instrument called the IncuSight Zoom. In the next slide, I'm going to show how we perform cardiac-directed differentiation to make hPSC-CMs and non-CMs. So we start with purified iPSC colonies at day -2, shown here in green. And we utilize small molecule-based approach for cardiac-directed differentiation, where on day 0 we use a Wnt signaling activator, followed by a Wnt signaling inhibitor on day 3. Then we switch to an RPMI-based media on day 7, and by day 9 of this process we obtain functional cardiomyocytes, and on day 30, after purification and maturation, we obtain very high purity and very lovely cardiomyocytes with rod-shaped morphology, and also with the appearance of multinucleation, which is characteristic of adult cardiomyocytes. We now generate these cardiomyocytes in T25 flasks, shown here, and using optical mapping, where we load the monolayer with a fluorescent dye, we can actually observe action potentials, shown in this figure. And early in the differentiation process, at day 17, the spontaneous beating activity of these cells is 3.4 hertz, which is the equivalent of a heart rate of 204 beats per minute. With time moving from day 17 to day 30, of the differentiation process, we observe slowing of the spontaneous beating rate from 3.4 hertz to 2.2 hertz, which correlates to a resting heart rate of 132 beats per minute. So there's some electrophysiological maturation that occurs just with time in these vessels. Now, when we generate these cardiomyocytes, we generate not only the muscle cells that perform the work of the heart, but also non-cardiomyocytes are formed. And it is important to be able to separate the cardiomyocytes from the non-cardiomyocytes, because the non-cardiomyocytes can have significant impact on the function of the cardiomyocytes. So here you see in the top panel spontaneous recordings of monolayers made up of purified myocytes. And when we slowly titrate in non-cardiomyocytes, there's significant speeding of the spontaneous rate of contraction, which gets even faster as the amount of non-muscle cells is introduced and mixed in with the cardiomyocytes. And this is shown nicely in the next figure, plotting the conduction velocity of the electrical impulse of the monolayers. And as we titrate in more and more non-cardiomyocytes, the conduction velocity slows. Similarly, whenever we quantify the spontaneous beating rate, as the amount of non-muscle cells increases in the monolayers, the spontaneous beating rate significantly increases. So that means it's very important to be able to separate and purify the cardiomyocytes from the non-cardiomyocytes. There are several approaches that have been developed to purify hPSC-derived cardiomyocytes. One of the first approaches utilized a mitochondrial dye combined with flow-activated cell sorting. This was developed in 2010. However, drawbacks of this method include low recovery of cardiomyocytes and risk of contamination. Next, a genetic modification and antibiotic selection approach was used. However, this type of genetic modification is not compatible with cellular therapy processes or with high throughput screening large numbers of unique patient-specific hPSC lines for personalized medicine. In 2011, a magnetic bead-based separation approach was developed utilizing cell surface markers, one called SIRPa 2A. This was reported in 2011. Another commonly used cell surface marker is called VCAM1, the vascular cell adhesion molecule 1. And while these were shown to be effective, with time the field has realized that actually these produce low viability of targeted cardiomyocytes and also low recovery of cardiomyocytes. And in our own lab, we found a high degree of user-dependent variability. More recently, a popular method utilized for purification of cardiomyocytes is metabolic stress selection. In 2013, a medium composed of high lactate and glucose depletion was shown to purify cardiomyocytes by essentially killing off the non-myocytes while the cardiomyocyte muscle cells survive. While this approach is very effective and very attractive because of its low cost and ease of use, the metabolic stress has unclear consequences for cardiomyocyte structure and function. So recently, in collaboration with Miltenyi Biotech, we've worked with them to create an innovative purification approach. And this is a new, highly efficient and clinically relevant approach which is needed for hPSC-CM purification. And this innovative approach targets the non-cardiomyocyte population for depletion by specific antibodies, and this consequently enriches the cardiomyocyte population. This slide shows a cartoon of this cell separation strategy for cardiomyocyte purification. The results that have been obtained both in the Miltenyi Biotech labs as well as my lab at the University of Michigan have indicated very high degree of purity, up to at least greater than 90%, independent of the pluripotent stem cell line used and the differentiation protocol and time points of differentiation. And this is a two-step strategy that's been developed. Strategy A involves depletion of the non-myocytes. So in this strategy, an antibody targets non-cardiomyocytes, shown here in green and in orange. Utilizing a magnetic field, these cells become magnetized and become trapped in a magnetic field, allowing the cardiomyocytes to flow through and to be significantly enriched. The second part of this strategy involves depletion of non-myocytes with subsequent cardiomyocyte enrichment using SIRPa 2 alpha antibodies to target the cardiomyocytes and to provide further enrichment of the cardiomyocyte population. This is a result obtained in the Miltenyi Biotech labs that's been shared with us, utilizing strategy A, depletion of non-targeted cells, or depletion of non-myocytes in this case. This shows data from flow cytometry experiments where cardiac troponin T is used as a marker for cardiomyocytes. Here we can see that in the unpurified cells there are two populations of cells. There are cardiomyocytes, which are cardiac TNT positive, as well as another population of cells which are making up the non-cardiomyocytes. With depletion of the non-myocytes, you can see that this population of cells that is the non-myocytes is no longer present at very significant abundance in the enriched cardiomyocyte fraction, indicating a very high purity of cardiomyocytes. In fact, in this case, the enrichment enriched the cardiomyocytes to 97% purity within the cardiac myocyte population. This slide shows a combination of using strategy A to deplete the non-myocytes and further enrichment using strategy B to further enrich cardiomyocytes. So in this slide, we see that the differentiated hPSCs contained just 19.49% cardiac TNT positive cells, and utilizing strategy A to deplete non-myocytes from the population, an enriched CM fraction was obtained up to a purity of 79.5%. And then subsequent use of strategy B for enrichment of cardiomyocytes resulted in purified cardiomyocyte population up to 94.3% purity. So you can see a stepwise improvement of the purification of the cardiomyocytes. So this is wonderful because it means that now, starting with cardiomyocyte ratios of 20 to 80% magnetic enrichment of hPSC-CMs using the PSC-CM isolation kit human gives purities up to 97%. In our own laboratory, we have tested strategy A, depletion of non-cardiomyocytes, and to date we have tested 8 different hPSC lines. Here, this is showing the results of flow cytometry analysis using cardiomyocytes differentiated from hPSCs before purification, In this case, the differentiation was of low efficiency of just 26.6% cTnT positive cells. And using strategy A, depletion of non-myocytes, we were able to increase the purity of the cardiomyocyte population to 84.9%. We've utilized other cell lines as well, here showing an hPSC line called 19911. In this case, the purity was a little bit better before purification at 68.4% cardiac TNT positive cells, and following strategy A, we were able to increase the purity of the cardiac TNT cells up to about 95% cardiomyocytes. And using a third control cell line called the BJ iPSC-CM, again, a purity of about 60%, we were able to increase the purity of the cardiomyocytes up to 96.3%, a very significant increase in the cardiomyocyte purification utilizing just strategy A. So next I'm going to show data where we went ahead and also utilized strategy B to further increase the enrichment of cardiomyocytes. So here shows with using flow cytometry to probe for cardiac TNT positive cardiomyocytes. The non-purified cardiomyocytes in this case were representing 30% of the cardiomyocyte population. We utilized an old approach using SIRPa, and we were able to increase this up to a purity of 50%. And when we utilized strategy A of the new kit, using non-myocyte depletion, we were able to increase the purity up to around 90% pure cardiomyocyte population. So using CM targeting directly with the old method using SIRPa fell short to provide purified cardiomyocyte populations, but strategy A on its own really improved the purity of the cardiomyocyte population. With further enrichment utilizing strategy A plus B, we didn't obtain any higher purity than using strategy A alone, which indicates that strategy A alone may be sufficient for enrichment of cardiomyocytes. We also used other cardiomyocyte markers, including MLC2A and MLC2V. These are cardiac myocyte-specific markers, and we saw a very similar trend where the non-purified cardiomyocytes represented 60% of the total cells initially. Using SIRPa alone, we gained no further enrichment. And using strategy A, however, we were able to obtain cardiomyocyte populations of greater than 90% purity. And with further enrichment by using a combination of strategy A and strategy B, there was tiny significant increase in purity, however, not reaching statistical significance. So it appears to us in our laboratory that utilizing just strategy A is sufficient to create very pure populations of human cardiomyocytes. Now I'm going to present data showing the structure and function of cardiomyocytes purified using the Miltenyi strategy A approach of non-cardiomyocyte depletion. So here in the upper left-hand corner, this is probing for cardiac Troponin T in red. This is a muscle protein, and in blue are the nuclei of each cell. And you can see there's very high purity of cardiomyocytes in this population under 10x objective using confocal microscopy. We also looked at the non-cardiomyocyte population just to make sure that we weren't losing any cardiomyocytes, and we found very little loss of cardiomyocytes in the non-cardiomyocyte population. Looking under higher magnification, using a 60x objective to get a better look at the structure of the cardiomyocytes, we could observe very good sarcomere striation patterns, as well as very good nuclear localization in the cells. And when we look at the non-cardiomyocyte population, we do see some cardiomyocytes that come through. However, the majority of the cells in the non-cardiomyocyte population are indeed non-cardiomyocytes. Here we've probed for a cell-to-cell junctional protein to make sure that the purified cardiomyocytes using this new approach from Miltenyi have very good cell-to-cell connections. Here we probe for the nuclei in blue, and we stain for N-cadherin, which is the cell-to-cell junction protein, in green. And what we can see is in between the cardiomyocytes, very good localization and very good expression of N-cadherin at the cell-to-cell junctions, indicating that following the purification approach, that the cells do what they're supposed to do, and they make very good tight junctions with one another. And here, using strategy A, we found that it is applicable to all of the hPSC lines that we've tested so far. At the time of making this slide, we had tested 11 different human cell lines, and all 11 cell lines were amenable to purification using this strategy. And here shows a beautiful picture showing the sarcomere structure, probing for cardiac troponin T, shown in pink, and N-cadherin in green. So we're very happy with this new approach of purification because it applies to many cell lines, every cell line that we've tested, and it has a very good workflow and only takes about an hour to complete. So in addition to performing structural analysis, we've also been performing electrophysiological assessments of these max purified monolayers. Utilizing just strategy A, the non-cardiomyocyte depletion approach. And so in our laboratory, we create monolayers, we load them with a voltage-sensitive dye called flow volts, and we perform multi-well optical mapping utilizing either 96 or 24-well plates. We use this dye to record action potential recordings. I'll show you data recording spontaneous activations of these monolayers, as well as data from electrical pacing. The electrical pacing that we use is shown here. We utilize a custom frame fitted with custom electrodes, and you can see in the center of every well a silver electrode that's used for point stimulation so that we can synchronize all of the wells and their contraction at the same time. So this is showing original recordings of action potentials. So here you can see everywhere that there's a flash of light. This indicates a spontaneous activation of the cardiac monolayer. And these cells purified using the Miltenyi approach have very good rhythm, have very normal rhythm, and importantly, when we do the same recording in the non-cardiomyocytes, which are made up of non-muscle cells, we do not observe any activations in any of the cells that we've used so far. If we look at the single pixel recordings, we can see very good rhythm again of hPSC-CMs purified using this new strategy from Miltenyi. And in the non-cardiomyocytes, since there's very minimal loss of the cardiomyocytes, there's absolutely no electrical activity in the non-myocyte population. We also have utilized this to measure electrical wave propagation. Here I show data, single pixel recordings from a single well of monolayers being paced at a frequency of 1 hertz. That's a rhythm of, or heart rate of 60 beats per minute. So this provides very good evidence for us that the new approach provides structurally healthy cells as well as functionally healthy cells. We've also utilized these cells to mature the phenotype of cardiomyocytes to enable high throughput 96-well plate screening for drug toxicity. We're able to actually control the maturation level of cardiomyocytes and make them either have a fetal-like morphology and phenotype or a mature morphology and phenotype. And that is shown here. Here, these are cardiomyocytes that have fetal phenotype. The cardiomyocytes are very circular in shape. However, the mature cells are much more rod-shaped and much larger. And that is quantified here, studying the circularity index. Mature cardiomyocytes are less circular, and the mature cardiomyocytes also are much larger. And when we look at the structure of the mature myocytes that can be obtained using the Miltenyi approach of purification, we can obtain cardiomyocytes with rod-shaped morphology and also with binucleation within the cells. In our 96-well plate mapping system, we utilize flow volts. This is the excitation and emission spectra of flow volts. The experimental setup that we use is shown in panel B. A 96-well plate of purified myocytes is put on top of a temperature controller, and utilizing a high-speed camera, we can obtain data from multiple wells simultaneously to enable the development of dose-response curves using purified cardiomyocytes. And at the moment, the array that we use is a 6x6 recording array, and this enables very high throughput and very high speed acquisition of data. So in this assay, in the presence of an action potential, the cells will fluoresce green, and we record this using a high-speed CCD camera. We have also utilized an analysis software system so that we can perform automated action potential detection. Here shows action potentials recorded from well A4, from this particular well here. And our software that we utilize labels the upstroke of the action potential in red and the repolarization phase of the action potential in green. So this enables very high content, very high throughput analysis of recorded data in an automated way. We validated the responsiveness of these cells purified using the new kit from Miltenyi, and we've exposed them to a compound that has low risk of developing arrhythmias called tamoxifen. And we've also utilized high-risk compounds which are known to prolong the action potential duration and cause arrhythmias. And here I'll show you data from a compound called Quinidine. So this shows different doses of Quinidine. The lowest dose is here at 0, and the highest dose used was 10 micromolar Quinidine, shown in the top row of action potential recordings. Each flash of light that you see corresponds to an action potential. And what you can see is the high doses of Quinidine. There's a slowing of the action potential firing rate, as well as a prolongation of the action potential duration. And that action potential duration in control conditions is about 350 milliseconds. And at the highest dose of this compound, which is known to prolong the QT interval and promote the development of arrhythmias, was prolonged to 537 milliseconds. On the other hand, when we used tamoxifen, which is a safe drug, we did not observe any significant slowing of the spontaneous beating rate, and we did not observe any significant increase of the action potential duration. So this indicates that the cells that are obtained using this purification approach respond to drugs as one would expect them to respond. So now I'm going to show you a case report from a study that we completed recently in collaboration with the FDA as part of the SIRPa initiative. And for this test, we were blinded. It was a blinded phase 2 study for the SIRPa initiative, and I'm going to show you the results for a compound that we now know its identity. The compound is called domperidone. It is a dopamine receptor 2 antagonist, and it is used clinically for Parkinson's patients to treat symptoms of nausea and vomiting. And what we found was quite remarkable, again using cells that are purified using the MAX technology. At the lowest dose used here relative to the clinical Cmax dose, at 1 Cmax, we saw very good rhythm of the cardiomyocytes. However, at dose 3, we could start to observe action potential duration prolongation, and excitingly, at the highest dose, we could actually see the induction of drug-induced arrhythmias that cardiologists must be careful of when they're using this D2 receptor antagonist. So at the lowest dose of cardiomyocytes purified using this new approach, we saw very good rhythm. However, at the highest dose, we could actually observe the induction of early after depolarizations that are known to lead to arrhythmias, and we could actually record the transition from somewhat normal rhythm to ventricular tachycardia-like activation of the cardiac monolayer. Now in my lab, we've been moving towards utilizing three-dimensional microtissues, and we found that for the development of three-dimensional cardiac organoids requires the presence of cardiomyocytes as well as non-cardiomyocytes. So the tissues that we use are shown in this picture. So in this there are 6 different tissues. The tissues form around 2 bendable posts. The posts are shown here as well as here. And we know from our own studies and the max purification technique that's been developed has allowed us to more quantitatively develop these. We know that in purified cardiomyocyte fractions there will be no tissue formation. The presence of the non-cardiomyocytes is essential for successful development of these three-dimensional tissues. And so now we're able to utilize this approach to recombine and make defined three-dimensional microtissues with defined populations of cells. These 3D microtissues are wonderful. They enable us to actually quantify organogenesis. So here we've developed an assay where cells are loaded with cell tracker green, and at time 0 the cells combine with collagen, take up the whole well, and over time the tissues form spontaneously through the work of the non-cardiomyocytes to work with the cardiomyocytes to reform the collagen matrix to enable generation of three-dimensional tissues. This shows the structure of those tissues. So here, standing for cell membranes with WGA, we can see that the cells are homogeneously distributed throughout the tissue. When we look at the structure, we can see very nice sarcomere structure being developed in the three-dimensional tissues. And when we probe for cardiac Troponin T, we can see that the cardiomyocytes are present homogeneously distributed throughout the three-dimensional tissue. And what's beautiful about this tissue is that we can use it to measure contraction, and here shows the actual measurement of excitation-contraction coupling. This shows calcium transient measurements in red, with simultaneous contraction measurements shown in blue. And we can see very clearly that the calcium transient precedes the contractile event in blue. So this enables a novel way to study excitation-contraction coupling in a three-dimensional bioengineered human tissue. So in conclusion, the StemMax iPS Brew stem cell media from Miltenyi which is xeno-free, is compatible with generation of virtually unlimited supply of patient-specific cardiomyocytes. The purified population of hPSC-CMs can be generated without genetic modification or metabolic stress using clinically relevant workflows, and we're very happy that the processing time to develop purified populations has been cut down to just 1 hour. Importantly, the purified hPSC-CMs using the new approach can be used for electrophysiology and for arrhythmia studies. And now we're able to manufacture 3D human cardiac microtissues. It can be done using clinically relevant workflow and, importantly, with defined cell populations. And with that, I thank you for your attention, and I'm happy to entertain any questions.
Thank you, Dr. Herron, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Herron will answer as many questions as time permits. And the first question is, do the purified cardiomyocytes freeze and thaw well?
Hello, yes. We've done experiments using the purified cardiomyocytes as well as non-myocytes, and we have frozen them and stored them in liquid nitrogen, and they do thaw very well. They come back to life. They can be used to reform monolayers as well as to create functional monolayers that have good electrophysiological function.
Can the purified cardiomyocytes be recombined with other cell types to create 3D tissues?
Yes, as I showed in the presentation, the separated cells can be recombined. They can even be recombined following freeze and thawing to create three-dimensional tissues, and we've done this using the platform that I showed in the webinar, and we also have used other types of vessels and other shapes, and they work very well.
Do you know the identity of the non-myocytes that are depleted from the cell population?
We're doing experiments to determine the identity of the non-cardiomyocytes. We know that they are very highly expressing a protein called vimentin, which is primarily a fibroblast protein. And also we've started to use some separation strategies to purify human cardiac fibroblasts using a similar type of approach based on magnetic bead separation. So those studies are still ongoing.
Okay, and as we wait for questions to come in, I would like to once again thank Dr. Herron for his presentation. Okay, here's another. Is there hope for using your cardiac cells to replace dysfunctional in vivo heart cells?
That is the ultimate hope for this technology, is to one day to be able to deliver healthy cells to an infarcted heart, to regenerate essentially dead regions of the heart. That's our ultimate goal. However, that will take quite a bit of research and quite a bit of work to be able to get there. But using this approach for the purification of the myocytes is a big step forward to enable defined cell populations to be used later on for cell therapies. Okay,
and additional questions are coming in. As they come in, I would like to thank you, Dr. Herron, for your presentation. Do you have any comments for us and any final comments?
The only final comment is that I'm happy to share this data in our endeavor to create clinically useful cell types. We think that using non-genetic modification of cells is a big step forward for the clinical use of these valuable cells that can be generated in a patient-specific way and hopefully to lead to personalized medicine and personalized cellular therapies.
Okay, and here's another for you. Can you recommend a publication or manuscript that details 3D human cardiac microtissues?
There have not been a lot of reports on the development of 3D microtissues with defined cell populations. However, I know that Dr. Murray's laboratory, I believe it was in around 2011 in the journal Circulation Research, his laboratory did report on the generation of three-dimensional heart tissue, I believe using human embryonic stem cell derived cardiac cells. Another leading group in the field is the group led by Dr. Eschenhagen from Germany, and his laboratory has really pioneered the generation of three-dimensional engineered heart tissue. They started back in the late 90s using rat neonatal myocytes to kind of figure out how to generate 3D tissues, and more recently they've been having publications come out, I believe, in the journal Stem Cell Reports, where they've described preliminary data generating human-engineered heart tissue.
For cardiomyocyte purification by strategy A, is it also applicable for isolation for mouse cardiomyocyte more than 90%?
We've not studied that in our laboratory. We've only studied human cardiomyocytes. I don't know that the same strategy can be applied to deplete the mouse non-myocytes, but there are a lot of similarities in the epitope expression between human cardiac cells and mouse cardiac cells, so it may be possible.
Have you compared your cells against other commercially available cells?
Yes, we've recently completed a study using our 96-well plate format where we utilized commercially available cells from Cellular Dynamics International called iCell cardiomyocytes, and we compared drug responsiveness between cells that we made here at the University of Michigan, purified using this new approach, and compared the responsiveness to the commercially available cells. And while the cells were similar, there were some aspects of the arrhythmia phenotype that were different between the two cell types.
How variable are the yields of cardiomyocytes?
So when we do a cardiac-directed differentiation of stem cells, there can be a pretty high degree of variability in the yield. As I showed in the webinar, sometimes we'll do a differentiation and obtain 20 to 30% cardiomyocytes. Other times we can obtain up to 90%. So the one very attractive aspect of the magnetic-based cell sorting is that we can rescue essentially a bad differentiation and still obtain highly enriched cardiomyocytes following the purification process.
Okay, well, thank you. Thanks again to Dr. Herron, and I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would also like to thank our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 7th, 2017. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.